Hello, and welcome to Virtual Investor Conferences. My name is Cecilia, and on behalf of OTC Markets, we're very pleased you've joined us for our next live presentation from Sigyn Therapeutics. Please note, you can submit questions in the box to the left of the slides. You can also view the company's availability for one-on-one meetings through the Schedule Meetings tab found on the conference platform. At this point, I'm very pleased to welcome Jim Joyce, co-founder, chairman, and CEO of Sigyn Therapeutics, which trades on the OTCQB venture market under the symbol SIGY. Welcome, Jim.
Thank you, Cecilia. I appreciate the introduction. Before I get started, I want to reference to today's participants our forward-looking statements that are also available on our website under the presentations button. And our website is sigyntherapeutics.com, and they're available through our regulatory filings at sec.gov. With that said, if you're not familiar with Sigyn Therapeutics, we create blood purification devices to overcome limitations of drug therapies. And some things you should know about our company. First off, we have a proven team whose members are involved in multiple first-in-industry achievements in our industry space. Previously, I was the founder of Athlon Medical, a company we grew from single shareholder startup to over 8,000 shareholders on NASDAQ, and a company that achieved multiple first-in-industry achievements. During our tenure, the market cap of the company reached or peaked at around $200 million. While I was there, I oversaw the development of the Athlon Hemopurifier, which we built from conception to the reality of it being the first FDA product to receive 2 breakthrough device designations, and subsequently it became the first technology of its kind to receive emergency use authorization to treat a pandemic virus. Subsequently to that, Time Magazine named the device a top 25 invention, as well as naming it to its 11 most remarkable advances in healthcare. I bring this up is because key members of our Athlon team, have now joined our Sigyn Therapeutics team. In terms of our pipeline, our lead product addresses significant unmet needs in global health. And then we have a pipeline that's comprised of devices to enhance the delivery of drug therapies. Our initial focus is based on oncology, but I can say that based on my 20 plus years in the extracorporeal blood purification industry, I believe we have the most expansive and compelling pipeline technologies in our industry space. In regards to our lead technology, it's called Sigyn Therapy. It's a broad spectrum hemoadsorption technology to treat pathogen-associated inflammatory disorders. Our technology is designed to treat both the pathogen causes of inflammation as well as inflammatory factors that can lead to organ failure and cause death. A little bit about this industry. The industry for hemoadsorption devices was pioneered by colleagues at Cytosorbents. They trade on NASDAQ under the symbol of CTSO. This is a good company. Their team is highly qualified. I participated with them for 5 years in a DARPA Department of Defense contract program to create devices to treat sepsis in wounded warfighters. Their Cytosorb device incorporates an adsorbent that depletes therapeutic targets that are below 5 nanometers in size, primarily inflammatory cytokines that underlie the cytokine storm. And their product has about 45,000 square meters of surface area. In regards to Sigyn Therapy, we incorporate multiple adsorbent components. We call it an adsorbent cocktail. Our adsorption area is about 200,000 square meters. That's the equivalent of about 50 acres of adsorption in one device. The device is highly efficient depending on the size of the patient. It has approximately 15 passes of the entire circulatory system through the bloodstream every 4 hours. And we're able to address therapeutic targets that are up to 200 nanometers in size. which is what allows us to address both pathogen sources and inflammatory factors simultaneously. Another aspect of our device is it works on the global infrastructure of dialysis machines already located in hospitals and clinics worldwide. This is an image of Sigyn therapy in action during animal studies at University of Michigan being deployed on a Fresenius dialysis machine. So I mentioned earlier about a device that could address both pathogen sources of inflammation as well as dampening down dysregulated cytokine production. A number of years ago, I was invited to testify before Congress related to the use of medical devices to treat emerging pandemic threats, emerging bioterror threats. And during my testimony, I stated there'd be a need for a device that could address a broad spectrum of viral pathogens, and that there would be a need for a device that could address the dysregulated inflammatory cytokine production that can lead to sepsis and underlies the cytokine storm. We designed a device that produces or performs both of those functions. And that is validated by preclinical in vitro studies demonstrating removal of a broad spectrum of inflammatory cytokines from human blood plasma, bacterial toxins, including endotoxin, viral pathogens, including COVID-19, hepatic toxins, and cytovesicles, which was a model study for removing extracellular vesicles that are transporting inflammatory cytokines. These targets just aren't circulating freely in the blood. They're also carried in these extracellular vesicles. So based on these outcomes, our technology is a candidate to address several significant unmet needs in global health. This includes sepsis, the number one cause of hospital deaths, endotoxemia, a leading trigger for sepsis, and a leading trigger for dysregulated cytokine production, pandemic viruses, which are happening more and more often based on global traveling, urban crowding, and global warming, drug-resistant bacterial infections, which are evolving to become a major issue, and community-acquired pneumonia, which is a leading cause of infectious disease deaths. Our next steps, based on our previous studies, is the filing of an IDE submission. This is an investigative device exemption submission to FDA. It's a modular feasibility study design. And what that means is we run a feasibility to demonstrate safety in human studies with our device, and then bridge off of that into pivotal studies for multiple indications. In this study, we're going to enroll 12 to 15 dialysis-dependent end-stage renal disease patients with endotoxemia and concurrent inflammation. A little bit about this market opportunity. There's about 550,000 of these patients in the United States each year that receive 85 million dialysis treatments. Once a patient is dialysis dependent, their average lifespan is 60 to 84 months, and endotoxemia and inflammation are a contributing factor to this increased mortality. And when we enroll patients into studies, previously at Athlon, we initiated and completed 5 different clinical studies under my watch. When we enroll patients, you like to know that you're going to be able to be efficient in your enrollment. So endotoxemia is reported to occur in about 40 to 90% of dialysis-dependent ESRD patients. The study protocol is going to be administer a device in series with regularly scheduled dialysis treatment sessions. So nothing changes in the normal patient's treatment schedule. Patient's going to be treated 3 times a week, 4 hours of treatment. There's going to be 6 total treatments. 3 of these treatments will be control treatments. Patients will be receiving their normal dialysis treatment. We'll be looking at blood chemistry factors before and after treatment. And then week 2, we will include Sigyn therapy in series with their dialysis device and quantify safety, but we'll also be quantifying changes in endotoxin levels and inflammatory cytokine levels. And it's really quite rare that you can run a control where a patient can be his own control in a feasibility safety study. But we think this is crucial so you can really monitor or quantify the difference in performance when the patient's receiving their dialysis treatment versus the dialysis treatment plus Sigyn therapy. Now, when we design our devices, whether it's Sigyn therapy or our pipeline devices, we design them with an intent that if they're demonstrated to be effective, that they'll establish a potential competitive advantage within an industry segment. And in this case, we feel that Sigyn therapy will do that in the dialysis industry. In the industry, the market is dominated by two companies here in North America, Fresenius Medical Care and DaVita Kidney Care. And what we've done is we've projected the revenue value appreciation for each month of extended life of Fresenius's and DaVita's patient populations. So in this regard for DaVita, each month of extended life for their patient population provides a value of $967 million based on 2022 revenues of $11.6 billion and a patient population of almost 200,000 people that they treat each year in North America. Now, this does not include revenue generation by keeping their patients out of the hospitals. When their dialysis patients end up in the hospital, which on average happens about 2 weeks a year, they lose revenues. And for each week of revenues for DaVita, they lose about 240 million across their patient population. So they can also increase revenues by lower levels of hospitalizations. We feel that if a company had access to this technology, they would take patients from other networks. And this does not include sales of Sigyn therapy to ESRD or non-ESRD markets. And for a dialysis company, this technology has the potential to pull them into the ICU, which would generate another revenue possibility. In regards to Fresenius, the potential revenue enhancement per month is even greater at $1.2 billion per month for each month of extended life for their patient population. And in this case, for each week of reduced hospitalization each year, that would add about 300 million in revenues. And again, some of the same factors underlying issues such as us capturing DaVita patients and being able to enter into the ICU to treat non-ESRD patients. So now I want to talk about our drug enhancement pipeline. And for those that aren't aware of our company or familiar with our company, this is the first presentation we've done in over a year. We've been very quiet and we've been quite focused on how we're going to enhance the benefit of drug therapies, especially those in the oncology space. So in October of last year, we filed a patent called Systems and Methods to Enhance Chemotherapy Delivery and Reduce Toxicity. This underlies two products, one named ChemoPrep and the other named ChemoPure. And I want to tell you why these products could be quite important. First, let's discuss the limitations of chemotherapy. As most of you listening know, it can be extremely toxic, and there's also high levels of chemo resistance that occurs over time. But what got my attention a little over a year ago in reading a report by some industry colleagues is that only about half of 1% to 3.5% of chemotherapy is delivered to its target tumor site. So the goal of ChemoPrep is to administer the device prior to the infusion of chemotherapy. Chemotherapy is administered in infusion centers, and we envision a device that will be administered in the infusion center to the patient prior to the infusion of chemotherapy with the goal to improve delivery of chemotherapy, an objective to reduce the dosing of chemotherapy by 50%, half a dose, which automatically would reduce toxicity by 50%, but actually achieve the same delivery, if not greater delivery. And this could be crucial because at present, and this has been an ongoing problem and is anticipated to be an ongoing problem going forward, there's a shortage of chemotherapy drugs, and physicians are forced to ration these drug therapies, which already are not being delivered very efficiently. And the targets that we're going after, I think you'll find this fascinating, are targets known as tumor-derived exosomes or tumor cancer exosomes. We call them tumor X's. They play multiple roles in cancer progression, but we are specifically looking at their role in blocking the delivery of chemotherapy. And this publication by Dr. Kolari at MD Anderson in Houston points out in cancer patients, their blood is estimated to contain about 4,000 trillion exosomes. That's a 4 with 15 zeros behind it. And based on an average size of an exosome being about 100 nanometers, that's about 248,000 miles of chemotherapy interference. It's quite substantial. And the rationale to deplete these targets is that they directly block and sequester chemotherapy delivery. They can actually export chemotherapy from tumor cells where the chemotherapy has been delivered, and they are a prime contributor to chemotherapy resistance. Post-infusion of chemotherapy is ChemoPure, and the use of this product is to deplete off-target chemo that wasn't delivered to the target tumor site and is still left circulating in the bloodstream. This is designed to further reduce toxicity for patients beyond the reduction by reducing doses required. And the one thing I will point out to some initial preclinical data related to this technology, the primary delivery mechanism are nanovesicles or nanoparticle delivery systems for chemotherapy delivery, primarily liposomes. So the removal of these chemotherapy agents, these liposomes, as you can see in this slide, we're able to substantially reduce these in an in vitro preclinical study. But these liposomes have long served as models for exosome removal based on size and the consistency or similarity in surface structures. So this is a very important data point for us going forward. Now I want to talk about the other product in our pipeline related to enhancing drug therapies. We call this Immune Prep. It underlies a patent that we filed in May. The patent's name was Devices for Enhancing the Activity of Therapeutic Antibodies. The limitation of therapeutic antibodies, similar to chemotherapy, they're poorly delivered. Less than 5% is delivered to their intended target, and they are often blocked by drug decoys. And antibodies have a tremendous specificity to bind to an antigen that resides on their therapeutic target. But unfortunately, there are many things that it can express that same antigen. The drugs themselves can produce antibodies known as anti-drug antibodies that will present that antigen and act as a decoy. There are circulating soluble proteins that represent that antigen structure that serve as a drug decoy. And then again, cancer exosomes or tumor X's also can cause a major problem. And in fact, if you look at this statement from this publication related to the use of rituximab in treating lymphoma, these researchers reported that exosomes had bound almost half of the dose of rituximab approximately 3 hours after the infusion of rituximab itself. And considering that the half-life of rituximab is somewhere between 72 to 76 hours, that binding mechanism was certainly going to continue to occur after that 3-hour data point. So again, just showing what a prominent role that these exosomes can have in blocking the delivery of therapeutic antibodies. So our solution with Immune Prep, we think it's quite novel. It's a reverse decoy mechanism. So essentially, we incorporate a therapeutic antibody within our device, and we circulate the bloodstream through the device prior to antibody infusion. And whatever binds to that antibody incorporated in our device would have been potentially a drug decoy that would have bound the antibody when it was infused subsequently into the patient. So decoy depletion clearly increases antibody availability to bind intended therapeutic targets. And we think this is a de-risk strategy. And I remember in my many occasions of meeting with reviewers at CDRH at FDA, I always recall reviewer saying, Jim, if you only incorporated a market-approved biological agent in your device, it would have a much better pathway. And there's no assurance of that, but we believe that incorporating market-approved antibodies that have already been demonstrated to be safe and effective will give us a regulatory advantage. And these are antibodies for which scalable production is already established. And we think this will be an attractive pathway that should get the attention of the drug industry and hopefully create some nice collaborative relationships. Our clinical opportunity at present, there's 162 market-approved antibodies. There's 1,200 therapeutic antibodies in clinical studies right now. 162 market-approved antibodies are expected to generate about $200 billion in revenues this year. By 2032, that number is supposed to exceed $600 billion. So in closing, we create blood purification devices that overcome limitations of drug therapies. And again, as I mentioned, when we design a device, we also focus on the device, if proven effective, that it'll create a competitive advantage in an established industry segment, such as the dialysis industry. Our corporate highlights, as mentioned, we have a proven team that's been involved in multiple first-in-industry achievements. Our lead product addresses significant unmet needs in global health, and our pipeline is a very expansive opportunity to enhance the benefit of drug therapies by improving their delivery. So near-term inflection points that you need to watch for related to Sigyn therapy are FDA IDE submission, disclosure of clinical site locations. At present, we're now evaluating 5 clinical sites based on our interactions with a dialysis industry leader who's helped us design our protocol. Look for an expansion of our management team, collaborative relationships, not just with our lead technology, but our pipeline technologies we think could drive a number of relationships. And related to our pipeline technologies will be in vitro studies that we anticipate we'll be reporting over the course of the next year. So with that said, here's my contact information, both my phone number, and if you want to reach out to me by email, it's the letters jj@sigyntherapeutics.com. And with that said, let me go to our screen. We have a number of minutes left. Bear with me while I navigate my Q&A screen. So I have a number of questions here. First question is, would Fresenius and/or DaVita be a potential partner, client, or source of capital? My answer to that is we know both organizations well. I established relationships with them in my previous company. We are working with DaVita on our clinical protocol. You'll see that one of our science advisors is the former global head of R&D for Fresenius. I think at the point where we're demonstrating that our technology is safe in human studies, then our discussions become quite serious. But I do believe we can create a significant competitive advantage for one of those companies, but we still have to demonstrate safety. The nice thing about our protocol and the safety study, we'll be able to not only quantify or monitor for reduction in endotoxemia, but we'll be looking at reduction of primary inflammatory cytokines and other factors related with inflammation. And again, we expect these patients, we're hoping for efficient enrollment, but based on the inflammation and endotoxemia suffered by these end-stage renal disease patients, we not only expect or hope to see reductions in levels of these targeting factors we're targeting, but we hope they'll feel better. So That's all I can say at this point on that question. Next question is related to, can I talk about our IP portfolio? Our IP portfolio related to Sigyn therapy is related to removing a broad spectrum of inflammatory factors in concert with removing pathogens and pathogen sources of inflammation, along with removing simultaneously extracellular vesicles that are transporting inflammatory cytokines. It's novel in the fact that it uses multiple adsorbent components. And then we have, and again, this is related to Sigyn therapy, and then we have a pipeline of some other submissions that we plan on filing to build more of a picket fence around the Sigyn therapy IP portfolio. As it relates to chemo prep and chemo pure, we filed in October of last year provisional patents that are very broad on this mechanism. It's not novel, the concept of removing chemotherapeutic agents, after chemotherapy to reduce toxicity, but it's very novel in our belief to eliminate exosomes or other circulating factors that are blocking the delivery as a means to reduce toxicity and improve delivery, and then to subsequently, as part of a system, further reduce toxicity by eliminating the chemotherapy that was off target. And at a certain point in time, once the chemo has not been delivered, it's more likely to cause toxicity than patient benefit. So the next question, in layperson terms, how does your chemo prep work? So I mentioned the substantial circulating levels of cancer exosomes. It's really quite amazing that that number of particles could be circulating in the bloodstream. Previously, in my previous company, we would address infectious viral pathogens where we were looking at hundreds of thousands of copies per milliliter of a virus or more. The copies per milliliter of exosomes in cancer patients is substantially beyond that. And the rate, something I didn't mention previously, the likelihood of chemotherapy resistance can correlate directly with the circulating quantity of these cancer exosomes. So first, we know that a lower level of these particles is going to be beneficial, but they're in such substantial quantities that any reduction, we believe, will increase the delivery of chemotherapy itself. And we believe, as I mentioned, our goal that with a half a dose of chemotherapy, our goal is to get equal, if not better, delivery. But the device itself is kind of a broad spectrum absorption device that really makes sure that we're trying to quickly, on a rapid real-time basis, sweep out these cancer exosomes that block delivery. Next question, and I have about a minute and a half left. Question is, can you discuss trial milestones over the next 12 months and capital position? So clinical milestones over the next 12 months, we expect to report a lot of in vitro study data related to our pipeline products. In terms of Sigyn therapy, we're going to be filing an IDE submission, expect to be disclosing clinical site locations. I can't assure, when, the IDE is finally approved by FDA, but there's going to be a discussion process, but we would expect over the next 12 months that we would have our clinical studies up and running. At present, based on discussions, we would anticipate 12 to 15 patients, as previously referenced, and that this would occur at likely 3 sites, which should, hopefully allow for initiation and completion of the studies within a period of 4 or 5 months. In terms of capital position, we've been funding the company as needed. We have some very supportive institutional investors and some angel investors, and we've been focused on the clinical aspect for the last year, and we now have a foundation under us where we believe we can be successful to go out to the capital markets and fund both our clinical endeavors and our other endeavors going forward. And with that,